Okay, so when we're talking about blood disorders or hematological disorders, we're talking about something that's gone wrong in the process of hematopoiesis or red blood cell recycling, or something's wrong with the way that our white blood cells are acting. So there's lots of pretty varied ways that you can have hematological problems, right? We're talking about clotting disorders. So I've kind of selected a small subset of these blood disorders as the things that I'd like to focus our attention to. And I've kind of grouped them as kind of things with erythrocytes, red blood cells, things with leukocytes, things like with white blood cells, and things that affect either equally. So this is, if you remember that chart where we showed like the family tree of how an original stem progenitor cell becomes all of these different kinds of cells, if it is arising only within the red blood cells, if some kind of error is occurring there, well, obviously it's only going to be a problem with our erythrocytes. So the first thing that I want to say is I'm not going to be talking about leukemia specifically in this lecture, because this is going to be like the homework to go out and watch a video of leukemia. So particularly I've chosen the chronic form of leukemia. I like this video because it actually talks a little bit about acute and versus chronic differences, but gives and chronic leukemia is a pretty common thing, so you're going to come across this in practice in the real world, unfortunately, fairly often. A Socratic question that obviously, again, everybody gets credit for. Everybody's probably heard of leukemia. Everybody's probably heard of sickle cell anemia. I would guess a fewer people have heard of hemophilia, if I'd have to guess. But this is kind of a little bit of a trick question, because thalassemia is actually the most common of all of these disorders. Thalassemia is something that we're going to talk about specifically. So when we're talking about how a red blood cell is supposed to function under normal circumstances, we've already talked about a lot of like how white blood cells function in the immune lecture, so I don't need to go too in depth on how that's supposed to work. But under normal function, a red blood cell is basically just a big giant bag of hemoglobin. There's not a whole lot else to what's going on inside of a red blood cell, these erythrocytes. This hemoglobin is actually not just one protein, but it's four proteins that are chained together into this macroprotein structure, where you have the heme group at the center of it, this little pigment group, and then at the very center of that pigment group, holding on to a molecule of iron. And iron is the star in this story, because iron is the actual thing that binds to this molecule of oxygen, right? This oxygen is lashed on to that molecule of iron. That's what's actually holding on to this. So when you look at each individual molecule of hemoglobin, it has the ability to lash onto four molecules of oxygen. Well, when you think that a red blood cell holds about 250 million hemoglobin molecules, we're talking about a billion or so molecules of oxygen within one red blood cell. So this is why red blood cells are so effective at their job. Their job is really, really important, so they're almost entirely devoted to making oxygen transport work and work well. When we're talking about hemoglobin specifically, most adults, when we're interacting with them, are going to be pretty much a mix of the alpha and beta chains, right? A hemoglobin molecule is two molecules of alpha globins and two molecules of beta globins, these protein chains that are linked together into this one hemoglobin molecule. At birth, we're actually more of a general blend between the alpha, gamma, and beta, though, right? Because there's a pretty significant difference in the types of environments that you're interacting with, right? If you are in utero and acquiring most of your oxygen at the placental barrier, the gamma version of this globin molecule is going to aid better in that. So as we shift to a more air interaction-based version of breathing, the beta is going to help us out. So that's why we see this kind of shift at birth. Hemoglobin are so devoted to their base function, right, carrying oxygen, that an individual erythrocyte is going to actually eject its nucleus, right? They shed their nuclei. Their final stage at which they kind of become an adult erythrocyte to go out into the world and do their job, there's this is a kind of a cost and a benefit. The cost we've talked about in the previous in pathophysiology, and specifically in anatomy and physiology 2, if you were to go back and talk about, hey, why is this a problem? Well, if you don't have a nucleus, you don't have protein expression. You don't have the ability to repair yourself. This is a big problem, right? Every time you bump up against a blood vessel wall, that's going to injure a little of this plasma membrane. So that's a problem. What benefit does this acquire? Well, twofold in that, hey, if I don't have a nucleus, a virus can't really touch me. The virus is not going to be able to infiltrate and create copies of itself if there's really no copier there, right? If there's no nucleus and no ribosome and transcription and translation mechanism within a red blood cell, who cares if that cell gets infected with a virus? It's not going to affect it at all. Additionally, just ejecting that nucleus means that now you have more room for this hemoglobin molecule. This hemoglobins are going to be less kind of jam-packed within this cell. So that's kind of cost benefit that we undergo. When we're talking about the three disorders that we're going to be talking about with red blood cells, erythrocytes, the first one of which is sickle cell anemia, and this is going to be an autosomal condition that is recessive. Autosomal is in that it is related to the first 22 chromosomes, so this is not going to be on the chromosomes X and Y, so this is going to be anything that's associated with chromosomes 1 through 22, is autosomal. This means that males and females are both equally likely to have this condition, and if you were to have a carrier, right, somebody that has one normal copy and one mutant copy, well, they have the propensity to produce both. If their kid gets their normal version, well, they might come out as normal depending on what the other parent provides. And if they get their mutant version, they might come out as the mutant expression depending on what their children are what the other parent is going to provide as well, right? So this is a pretty common carrier state. And so when you're talking about groups that have high incidence of sickle cell anemia, knowing the carrier status of both parents can inform a little bit about what things to expect with the offspring. In sickle cell anemia, beta globins are the thing that's actually wrong. The protein that is defective in this case is not the alpha chain, but it's the beta chain. So remember, hemoglobin is really 4 proteins that are lashed together. So if 2 of these have errors in them, it's going to change the way that this hemoglobin molecule works. This is a pretty minor effect, right? So when we're talking about what's actually going on inside this hemoglobin molecule, a mutated version accepts an oxygen molecule, right? It's going to operate relatively normally. The diagram that you're seeing here is it's just one minor base change in that it's replacing a glutamate with a valine molecule amino acid. So now the protein is going to have almost everything the same except that one particular amino site. So this means that this hemoglobin molecule, yes, it can hold on to oxygen, but in the event that this hemoglobin lets go of its oxygen, well, now that hemoglobin molecule changes its shape a little bit. And this becomes a big problem because now this new hemoglobin that's been deoxygenated, it's lost its oxygen chain, is actually going to start linking up with other deoxygenated versions of hemoglobin. And you start forming this kind of long rod of this clumped hemoglobin. The beta chains are going to stick to each other in this case. And it forms this long rod. And what happens when you have basically a pencil, a long rod growing within a balloon, right? If you could imagine this hemoglobin being a rod that's extending itself, as this molecule becomes deoxygenated, it gets stickier and the rod gets longer and longer and longer. Well, that's going to cause this event where this long rod causes a misshape in the actual erythrocyte. It's going to cause this kind of half-moon crescent shape, or this sickle cell shape, right? I'm trying to say that 5 times fast. This sickling is actually the extension of this hemoglobin rod within this red blood cell. And it has to do specifically with what happens when you let go of a molecule of oxygen. This is not great, obviously. If you would imagine a pencil growing inside of a cell that does not have the ability to repair itself, that's obviously going to injure the cell, and it's going to make it more likely to undergo hemolysis. So if we are entering into areas that are already low oxygen, well, that's going to make that rod go faster, and then those areas are going to be more likely for the red blood cell to rupture. So think the hands and feet, right? The further you out, the more less blood that is being delivered to those areas, the less oxygen that's being delivered. So the hands and feet are going to be the sites of majority of this hemolysis that's occurring. Remember, heme is not something that we ever want to actually release out in the system. Heme is something that is really, really toxic. It's really damaging. That's why we have a spleen. The spleen is there to capture these red blood cells that are kind of about to go bad, about to pop, before they pop out in the vasculature system. So when we have a bunch of hemolysis occurring without in the systemic circulation, that's a really big problem. It's going to cause a lot of toxicity. Luckily, we have a molecule called haptoglobin. Haptoglobin is hanging out in circulation and is kind of sucking up all of this free heme, right? When heme is released, haptoglobin is going to bind to this heme molecules and prevent it from doing too much damage. So this is kind of a temporary pause on the amount of damage this heme can do if we have this red blood cell burst out in the fingers or out in the hands. Eventually, this haptoglobin makes its way back to the spleen, and it gets digested in the same way that normal red blood cells get digested by a macrophage. Then it undergoes the normal step-by-step process, right? The hemoglobin is then digested, eventually into unconjugated and then conjugated bilirubin, and then excreted as either in the feces, urine, or in bile, right? That's how things are supposed to go. If we have sickle cell anemia occurring, well, all of a sudden, way more of this is occurring. And not only is this going to cause problems with increased rates of hemolysis, but also the sickled cells, these cells that are kind of extended chains of hemoglobin molecules because of deoxygenation, right, letting go a molecule of oxygen. This is going to kind of cause these sickled red blood cells are going to be less likely to flow easily through circulation. They're going to cause more clumps. They're going to get into traffic jams. When this happens, specifically in the smaller capillaries as they're moving out into the tissues that they're actually delivering the oxygen to, this is going to cause a backup, and this causes occlusion or vaso-occlusion crisis. VOCs is a pretty big deal, right? When this is happening inside the lungs, it's going to prevent the blood delivery to various alveoli, and then you're not going to get that blood oxygenated. If this is occurring in the brain, obviously that's a really big deal because now we have a potential infarction occurring. If this is occurring in the hands and feet, this is going to affect things like growth, and potentially bone death is going to occur at greater rates. So this is why it's not only the hemolysis is a problem, but certainly also the shape of even these unburst versions of red blood cells is also a big deal. What are you going to present when you have sickle cell anemia? Well, you're going to have anemia because those red blood cells are going to be bursting too fast. They're going to be exploding out in circulation before their life cycle is too short. You're going to have hyperbilirubinemia. Bilirubin, remember, is part of that metabolic process where we take that heme molecule, cleave off the more damaging portions of it, and convert it into conjugated or unconjugated bilirubin. Well, if you have way too much hemolysis happening, there's going to be way too much bilirubin accumulating, and the liver can only process so much at any one time. And the vaso-occlusion crises are going to cause a lot of problems because they're going to have infarcts occurring in different areas around the body. Wherever they just happen to get gummed up, you're going to see ischemia in the kidneys, in the liver, in the bones, and it becomes a big problem when we're talking about neural involvement, right? Obviously, the brain is not one of these things that we want to allow any tissue to die. This is also why priapism is something that's common in sickle cell anemia patients. Priapism is you acquire an erection, right? Males acquire erection, and then because of a blockage in the drainness of the penis, you actually can't ever make that erection go away. And it causes a problem because it stalls the blood within the penis itself. Eventually, that blood is going to use up all of its latent oxygen, and the penis and supporting structures are going to die because of the fact that the blood that's pooled in them and not really moving and not ever leaving them eventually loses all of its oxygen. So this is a big problem. This is also why you see hand-foot syndrome in patients with sickle cell when you're a kid. Hand-foot syndrome is, hey, there's a bunch of bone ischemia and potential infarctions that are occurring, right? If you have a lot of deoxygenation occurring and hemolysis occurring out in the hands and out in the feet, that's going to eventually damage the hands, and it's going to cause these hands to become stunted in sickle cell anemia patients. Because of the fact that this is the areas that most deoxygenation is occurring. Why does this happen in the first place? It's pretty simple to understand because sickle cell anemia, fortunately/unfortunately, provides protection against malaria. If you have sickle cell anemia, if you're a heterozygote or a homozygote, so if you have one mutant copy or you have two mutant copies and have sickle cell anemia, the heterozygotes generally don't express that much disease. If you are a heterozygote, you are still protected against malaria. Malaria is a Plasmodium falciparum, this little tiny this little tiny pathogen that makes its way into red blood cells. Well, if you have the sickle cell trait, it actually protects and prevents this malarial Plasmodium from getting into your red blood cells. So if you were to draw a map of where malaria is versus where sickle cell anemia is, it's pretty much a 1-to-1, right? It's a pretty clear distinction that, hey, there's areas that you have lots of malaria. It's a pretty common selection force for this is showing up in the human population. We actually think that sickle cell anemia might have evolved a couple times. So that's why you see like a Arabian Peninsula and an Indian subcontinent version of this, as well as a sub-Saharan Africa. A question, why are you not concerned about sickle cell anemia, especially even if somebody that is a homozygote, why are you not concerned about that until after birth? Well, it's not because of fetuses don't have a high oxygen requirement. That's not true, right? They have a bunch of oxygen requirement. They're growing. And it's not to do with alpha because alpha is fairly constant within life. And it's not really to do with malaria because malaria is the thing that sickle cell protects you against, but sickle cell can still damage you if you're a homozygote. It is the fact that beta globin production only really starts to pick up after birth, right? Prior to that, we use mostly alpha and globin production, alpha and gamma globin production, rather than alpha and beta globin production. That is the distinction. Thalassemia is a similar condition when you're talking about the levels of hemolysis that are occurring. And it's actually one of these things that is pretty understudied compared to something like sickle cell anemia. We have a lot of people with sickle cell anemia in the U.S., but there's actually way more with thalassemia. Part of the reason is like who has thalassemia. Thalassemia is pretty common around the Mediterranean and throughout Southeast Asia, so it's not as common here in the U.S. Additionally, thalassemia is not going to cause the same sort of really, really negative effects as sickle cell, so that's why it's a little bit less well studied. There are actually two distinct forms of thalassemia. There's alpha and beta thalassemia, and if you have been tracking along, alpha and beta refers to the globin molecule that is actually causing the problem. So you can think about alpha thalassemia don't have enough alpha hemoglobins. Beta thalassemia don't have enough beta, right? So the the flip side of this is there's too much beta or there's too much alpha hemoglobins. Both of these cause problems with hemoglobin, with red blood cells, because of the fact that if you have a alpha and a beta and an alpha and a beta chain, they're going to form together in this kind of Voltron, kind of multi-part organ that comes together, right? This is how things are supposed to work. But what happens is if you're making way too much alpha or you're making way too much beta globin, well, all of a sudden there's not really anything for them to stick to, and they just kind of hang out in that red blood cell, eventually precipitating out to the plasma membrane. They fall out of solution, and then as they precipitate, they irritate that plasma membrane, damaging that plasma membrane and making it more likely to rupture because we don't have repair structures. Remember, red blood cells don't have a nucleus. So I'm going to have a little drawing on this. Right? Oops, other drawing. Spoilers. So if we're talking about alpha thalassemia or beta thalassemia, the difference is only how much of the one particular version of this hemoglobin molecule that you have. Under normal circumstances, you have one molecule of alpha bound to another molecule of alpha, bound to another molecule of beta, bound to another molecule of beta, right? So 2 alphas and 2 betas. I'm just going to draw it. I'm not going to use green because I'm going to try and be cognizant of our colorblind individuals. So alpha, a normal molecule of hemoglobin is 2 parts beta, 2 parts alpha. In alpha thalassemia, the problem is you don't make enough alpha hemoglobin, right? So when you end up having these molecules, you end up having some molecules that are bound, right? So someone with alpha thalassemia still has hemoglobin, right? Same thing where you have a patient that has beta thalassemia. You still have hemoglobin in this patient. But the problem is you end up making a unbalanced amount of one of these molecules, these proteins, either the alpha or the beta version of this. So in alpha thalassemia, you make some alpha, but you don't make enough. You don't make equal amounts alpha to beta. So you end up producing more of this beta. So this beta globin, this beta protein, ends up hanging out and doesn't really attach to anything, and eventually it starts sticking to the walls, and by sticking to the walls, it causes a little bit of irritation, and this red blood cell is more likely to rupture. The exact opposite case is true in beta thalassemia. The beta thalassemia, the problem is you're not making enough beta globin. You have more alpha globin that's unpaired, and eventually this alpha globin is going to precipitate out and start sticking to the walls and causing problems. So that's, in a very, very small nutshell, what's going on in thalassemia. So this is going to look an awful lot like sickle cell, right? Increased rates of hemolysis. That increased hemolysis is going to lead to the spleen working hard to try and keep up with this and ends up damaging the spleen. And because the spleen kind of ends up being outpaced by the degree of red blood cell destruction, you end up having a lower red blood cell count than other individuals, so you're anemic. Because you have this occurring in pretty much everywhere in the body, these bone deformities are more likely to happen because bone growth requires a lot of oxygenation and a lot of nutrient delivery. Thalassemia is a little bit more complicated than something like sickle cell because there's big degrees even within alpha or versus beta thalassemia. There's some individuals with beta thalassemia that's a very mild form, and there's some individuals with beta thalassemia that's a really pronounced, very deadly form of thalassemia. So it is a lot of variation within this that we don't really have time to necessarily go into. The reason for thalassemia existing is the fact that we have some general protection against malaria, right? Alpha thalassemias are, again, in the picture you can see it's spread throughout generally the same regions. Mostly when we're talking about alpha thalassemia, we're talking about Mediterranean, a North African. Mostly when we're talking about beta thalassemia, that ends up being through like India and other areas of Southeast Asia. So it's spread across both regions, but the generalities is Mediterranean versus Southeast Asian. So that's not necessarily a complete one-to-one though. Well, thalassemia and sickle cell anemia are very, very similar conditions because they're both causing these misshapen red blood cells, right? They end up damaging they occlude vessels and block vessels, as well as popping and release their heme molecules, ends up damaging the red bloods, damaging the vasculature. Well, how do you deal with this? You're anemic. Well, you just get a blood transfusion, right? If you don't have enough red blood cells, if you're anemic, you just get a red blood cell donation from another person. The problem is, if you have thalassemia and if you have a bad case of thalassemia or a bad case of sickle cell anemia, and you're getting blood transfusions once every 3 weeks, right? It will be a pretty high rate of blood transfusion, right? Going into the doctor and getting new doses of blood transfusion every time someone is giving you someone else's red blood cells. This becomes a problem because now, remember, if I'm bleeding and if I have like a big giant gash in my arm and the blood is splurting out, I'm losing that red blood cell. I'm losing all of the heme and I'm losing all of the iron that's going with it. So if I get a blood transfusion, that brings me back up to my normal iron concentration. If I have thalassemia, well, thalassemic individuals are going to have hemolysis occurring, so they don't have red blood cells working properly, but most of that iron is going to be recaptured when that red blood cell pops, right? It's mostly going to be recycled. So if I then get a blood transfusion to bring my red blood cell count back up, well, now I'm adding more iron when really I wasn't actually ever losing it. So now I have to be cognizant of the fact that I might be iron overloading this patient. A little bit on iron metabolism. Iron is incredibly consequential in red blood cell in erythropoiesis. Iron, this is the vast majority of the iron in our body, ends up being devoted to this process of red blood cell production. We end up being, under normal circumstances, really stable in iron. We absorb a very small amount in our day-to-day consumption, and we end up defecating out a very small amount of it. Well, in normal circumstances, we don't really have the ability to, if maybe I lose a bunch of blood, I don't have the ability to just increase my iron absorption, right? Some of that is being replaced, but the problem is if I have way too much iron being added to the system, things like a red blood cell donation, I can't really excrete it, right? I can't get rid of it all that well. I can store some of this for a rainy day in molecules like ferritin, but there's actually only so large of only so significant of an effect that I can actually have on this. So when I am someone that's receiving consistent red blood cell donations, I have to be concerned about things like hemochromatosis. Hemo as in blood, chroma as in a metal, right? There's too much iron, and it ends up depositing in areas like the liver and in our bones, in our skin. When this is depositing in the brain, it can accrue in the pituitary gland. Remember, the anterior pituitary gland is sending out a lot of signals, and a lot of times when we're concerned about patients that are receiving consistent iron donations, red blood cell donations, we could be damaging the pituitary and causing Addison's-like syndromes, right? So something that we've already previously covered. This becomes a problem because these blood transfusions end up adding more red blood cells, which then will eventually be broken down, right? They're not going to be as damaged as the red blood cells that we have in someone with thalassemia or sickle cell anemia, right? Because they don't have the same mutations in those individuals that are donating the blood. But as this iron starts to increase in concentration, now we start looking for places to shove all this excess iron. So we start putting it into our organs, we start putting it into the pancreas, we start putting it into our skin, right? Particularly places like the liver and the heart tend to get hit pretty bad. But we could have someone that has sickle cell anemia develop diabetes because the pancreas ends up having this so much iron precipitated and ends up being slightly toxic to the pancreas itself. A pretty characteristic thing is as the skin starts to have more iron deposited, it starts to look almost kind of bronze, almost like you have a really weird off fake tan, right? That is because of excess iron that's showing up. That's what you're seeing in this patient. How do you deal with iron overload? Well, the way that you deal with iron overload is you remove the iron. The problem is it's really hard to grab and remove just the iron without causing a further anemic damage, right? I don't want to just blow a bunch of red blood cells and cause them to be excreted. That's a big problem, right? That's not something that I ever want to actually have happen. So we end up giving individuals chelation treatments because these chelation literally means to claw or to grab. So these chelating agents grab onto these iron ions and they allow them to be processed out by the kidneys. So the kidney would basically see this iron ion and skip over it, right? It completely ignore this. I don't have the ability to excrete iron ions through my kidneys. But this metal chelate molecule, well, this is actually going to be processed by the kidneys and ends up being deposited in urine, right? So that's the big difference in this. We're going to stop there because we're going to talk about hemophilia and we're going to talk about paroxysmal nocturnal hemoglobinuria on Monday of next week. So I will stop here and